The next speaker we have is Xiaoxi. Xiaoxi is an award-winning entrepreneur, and she is the co-founder and CEO of a company called Xtherma. So when I was describing their company on the website, I used the word cryopreservation, and she immediately corrected it, and she said, she used the very apt word, biopreservation. We are at the hub of this organ and tissue engineering laboratory, and I think her technology fits here very well. If our goal is to have tissue replacement, organ replacement, and that represents the future, the technology that Xiaoxi is going to describe, which is enabling survival of organs outside of the body for extended periods of time, I think this could definitely be a part of the future we are all looking forward to. Over to Xiaoxi.
Thank you so much, Josie and Aubrey and Bernie, to invite me to share some of what we have done and to the world leading thought leaders in stem cell biology and all the therapies. So you can see here, everything listed on the screen is a living medicine. So they're very different from our traditional chemical pills. So if we just put them on the bench top and do nothing with them, they will die out with a few minutes because they're living. So Xtherma is a company really laser focused on actually advance the biopreservation technology and bring up the new platform so we can enable all those living medicines become off the shelf. To start this journey, actually, it's a very strong personal inspiration. I unfortunately lost my grandpa from not getting a liver for transplant on time. So as an 8-year-old, that was very difficult for me. And my mom, as a physician, told me all about transplant. It's not because there's no donors, it's really we don't have enough time. So that really seeded me, tried to become a biohacker. And until in the college time, I studied biotechnology, I learned low temperature indeed can make this happen. So since then, I pursued my PhD in chemistry and a start company in 2014, right after my PhD. So now I'm very grateful be able to still getting on my dream route. So we grow the team of 30 and headquartered in the San Francisco Bay Area. And we also have a first subsidiary company in Austria, Innsbruck, a very beautiful place where we do a lot of engineering work. So we actually grown really fast thanks to the Lawrence Berkeley National Labs a cornerstone help, giving us this state-of-art facility, also the founding father of this type of technology, and we can work day-to-day basis. And beyond that, we're grateful to receive over $14 million non-dilutive fundings to date, so led us to laser focus on the science, and before we went to the commercial route. And now Xtherma is a post-Series A company. Actually, we're very excited to first receive FDA's breakthrough designations, potentially completely transform how we do organ transplant. So you can see today, even decades later, organ shortage is still a main issue. So you can see in US, actually, we have more than 10 times the people actually waiting for organs. Sadly, every single hour, we lose one life on the wait list. You may think we don't have enough donors. That's not entirely true. More than 150 million of US citizens register for donation. One donor can save 8 lives. But the shocking number is here we waste over 80% of the organs. Why? Actually, the main reason here is we just do not have enough time. Because our poor preservation technology we use today, since 1950s, we've been using the same way. When organ become available, we'll use a sugar salt solution, put on the white ice, ship it in the igloo cooler. This way, heart only lasts for 4 hours, kidney 16 hours. That's why you heard those stories, the organ die in the winter storms, stuck in the airport, all those issues. So we really have to get more time for the surgeons to do their best and to save patients' life. So Exterma really asks a very fundamental question. What if regenerative medicines can be frozen without a freezer burn? You know, like cells, tissues, and organs. What if we can play a little game with the temperature? So obviously, like everything in your freezer lasts longer than refrigerator because the temperature is lower. But there's one problem. You don't want to have a freezer burn. You never see a heart come out of the freezer actually come back to beat again. And you have to add some molecules to prevent the freezer burn. So can car coolant work? Obviously, you cannot put the car coolant into the organs, expect them coming back. Also, DMSO does not work. We use 10% DMSO to preserve isolated cells. It works okay for 50% coming back, but you just cannot implant 50% workable heart. Also, DMSO has been extremely toxic to bring the organs back because the amount we have to use here. So the answer is we really need to look for new molecules, not only organs. If you look here, every biologics, every regenerative medicines from the cancer tumor cells and the apheresis raw materials, stem cells, also to the kidney for transplant, everything has their own time. If we ship fresh and then preserve at 4 degrees C. So what if we can just dive down this temperature a little bit, then we can technically extend this curve to the right with the same amount of time. You get a better quality of the product. With the same quality, you have extended the time. But here all comes down to the challenge. We need new molecules. DMSO, car coolant cannot fulfill this goal. We need to look around. So where can we find it? Albert Einstein said, if you look deeply into the nature, you will understand everything better. So that's what we did. Exterma take a completely different route. Actually, going to learn what nature does the best. You can see there are fish and insects, and they live in the Arctic Ocean. They do not use the liquid nitrogen to preserve them, and they do not go down to that kind of temperature. Instead, they chose a very mild way, and by creating those special proteins only when available in the winter time for very small quantity. And it's called antifreeze protein. So, but the antifreeze protein cannot be upscaled for human use and also bring a lot of scale-up issues. So Exterma become the world's first company be able to dissect it into the molecular design principles and build a rational design library, goes through robotic synthesis, and the AI-guided screening process truly use the modern drug discovery platform, goes through in vitro, ex vivo, in vivo transplant to confirm our finding. So this molecule, actually named the peptoid, you can see by just shifting the carbon, the side chain from the carbon into the nitrogen, and you get something extremely similar to peptide, but immediately solve the mismatch issues. Actually making them immediately scalable for medical use and does not cause immunogenicity issues, stability issues. So the image on the right not only get us some prestige awards nationwide for nano art contest, really this shows the power of the chemistry. So the top one, actually this is under the polarized light. The water, if you shine the polarized light on top of the ice crystal, you'll see this colorful display. And that's real color because ice is a crystal. You can see the top one actually coming from water. If you bring after party ice cream back to home, back to the freezer next day, it never tastes good because the ice just like to expand bigger. But the bottom one, you can see this is from fish antifreeze protein. Immediately, you notice smaller ice crystal sizes. But actually, for XTV, for our molecules, even with a reduced concentration compared to the fish protein, you get rounder and nicer crystals. More importantly, we use such a low concentration, we can prevent this kinetic process and prevent the ice to merge bigger and get bigger. So this really holds a secret. Now we can develop this turnkey solutions, ex vivo. So this is a new type of chemistry, and we're able to build this first time DMSO-free, protein-free, chemically defined biopreservation solution that now can preserve biologics below 0 degrees C, above -20 degrees C, with ice-free condition. And at the same time, actually, we built a smart transporter called TimeSeal. TimeSeal is engineered in Europe. Like, we bring the whole team together by talking to transplant surgeons, by talking to process engineers from the cell therapy companies. We built this turnkey solutions, passive system. You don't need to have active cooling, and you have the enabling solution. With the combination of such, you can totally remove time out of the equation for the regenerative medicine, and can cover from the raw materials, and think about the vaccines, and to the tissue engineering construct, all the way for organ transplant. So we have been luckily validated the technologies through the cells, tissues, and the organs, and all you can see like a therapeutic type of cells from 20+ partners, thanks to our robust grant system as well as our early access commercial program. So here I would like to share with you some of data we have. So, ready to use organs on chip or tumors on chip has been a dream in this field. But the problem is like all of those difficult 3D printed growth construct has extremely short life to live. If you use a traditional cryopreservation method, they do not last. So, for example, when the CERN first found us, like actually Glasgow Institute researchers are ready to bring the iPSC-derived cardiomyocytes back to the clinical trial. But the problem is, every time when they freeze down this 35 days derived cardiomyocytes, they lose 85% of the cells. And the rest of 15% take more than 3 days to come back to beat. When they beat, they don't beat like the heart cells. So, with this method, we showed just by growing the cells in the petri dish, put into the TimeSeal, pour ex vivo on top, 5 days later, you can see viability being well preserved. And more importantly, we can preserve the heart function after you giving like a calcium pulse and the heart start beating, just like a regular way. And then we've been receiving like some grants from BioFab USA, the consortium doing tissue engineering, and we're helping 3 of the portfolio companies. Each one of them are working on some really cutting-edge, complete tissue engineering system, tissue therapies. But one example here, you can see Theradaptive, like they're doing this organoid on chip. And this is the human primary liver organoid. You can see we developed from like a 5K all the way to 80K. It's really large clusters. And using this TimeSeal ex vivo method -5 degrees C, we're able to preserve for 1 week long, and all those clusters remain intact and similar functions to the fresh ones. And then we did some control studies. Not only you can see on the top ones, not only ex vivo remain the same viability as the control, the fresh samples versus the traditional hypothermal, so 4 degrees C preserved one actually has low viabilities. Also on the bottom you can see for the liver functions we're able to produce healthy amounts of albumin compared to the fresh, never frozen ones. And we've also been funded by NSF. Like here, we went into the full thickness skin preservation. Compared to the traditional glycerol ringer solution of 4 degrees C, I think like the display is not super good here. But after 14 days long, we checked the full thickness skins, and the entire skin constructs have remained really good. Integrity, histology data, and also shows very low apoptosis compared to the control, as well as shows healthy proliferation potentials compared to the control. We're also stepping into the neuron world. So ExxonMobil developed like cutting-edge therapies to treat the wounded warriors, giving them regenerated neurons. But this is among the most difficult to preserve the system. I believe many of you already know. But here you can see after 2 weeks long, we call it short-term preservation, but 2 weeks long, the neuron graft actually remain completely alive compared to the control is completely dead. Just like the difference between using XTV for -5C versus the 4 degrees C is normal. And we've been working with other collaborators on the MSC-derived chondrocyte construct. And this very large system, actually, we now prove that we can do 6 months preservation. Still, you can see remain very good viability, and all this collagen type of lineage, like markers. So this is really solve those shelf life issues for those tissue engineering products. Of course, the most exciting thing we have achieved is on organ transplant side. You know, like for human hearts, or any heart, it's among the most sensitive type of organs and only have less than 4 hours. And here, actually, we work with the leading transplant team in Johns Hopkins, Dr. Gary Brandeker's group, showing like black and white results. When I first brought my data to Dr. Brandeker, he said, "Oh, sure, yeah, it works." But it just sounds too good to be true. What if we try something really challenging? We know 24 hours, there is no method, no matter what you use, to bring a heart come back to beat. So we actually did a head-to-head comparison using one side is HTK, what the organ transplant gold standard at the 4 degrees Celsius working temperature above 0. 24 hours, no magic happen, all the heart is dead. You can see this is a post-transplant as soon as you release the clamp. On the right is the HTK group. On the left is the ex vivo and time seal group, -5C, 24 hours. The heart remains fresh, and as soon as you release the clamp, the transplant resume, and the blood flush in, and the heart come back to beat within 2 minutes. And we not only done it once, we have done it dozens of times. And so we also, thank you. And then we follow up with all the post-transplant analysis, not only to see the heart come back to beat, we also check all the apoptosis markers and the gene expressions, shows indeed like this method slow down the ischemia damage and be able to keep the organs alive and the mouse also alive. And then some of our substance actually survive them more than 2 years, like the normal lifespan. So we did not stop on the mouse heart. Actually, we immediately go into the kidney side. So as soon as the Hopkins opened up in 2020, and I received a phone call from Dr. Brandeker, he said, "It worked, it peed." I said, "What do you mean? Tell me more." He said, "I transplant, I actually took one of the kidney out from the pig, and then I preserved with XTV for 48 hours. And then immediately we do this. You can see here, I don't know if this play. So this, after 120 of kidney recovery, and as soon as you're giving like a warm blood perfusion back, this kidney turn pink. This is complete transplantable conditions. And the most important thing, as you can see, immediately generating a clear urine productions. That means that the kidney not only look good, also function right. So again, we're not only done it once, we actually done more than 40 plus pig studies. So based on those data, actually, we'll be able to submit it to FDA, and FDA grant us the breakthrough designations, potentially first indication in human kidney transplant. And we can turn traditionally 16 hours to potentially 120 hours. That is 5 days. Not only we can immediately solve the global organ shortage issues And also, we can think about bring the kidney to the moon and bring back, still can transplant. Thank you. Yeah, we're excited to have this on the way to solve the shortage issues. I'm sure everybody heard about this new xenon transplant breakthrough last year. And the MIT Review just write an article about when we can see the organs on demand happening, probably another 10 to 15 years. And now here, we have a tissue engineering hub. We're working on lots of 3D printed tissues, 3D printed organs. We see Extherma so here really wants to support all those endeavors. So Dr. Brandaker said, the beauty here using this system, because we focus on so much on the fundamental level improvements, actually, we do not change the peri-transplant workflow. I got to learn a lot of amazing transplant surgeons. I see how intense this work can be. So we actually maintain this whole system exactly how you've been trained for traditional transplant. Instead of just using the UW solution, sugar salt solution, and the igloo cooler, now you swap them into the XT Vivo and time seal. You don't need to change anything else, and you got the extension of the time and better organs. Oh, sorry. Yeah, and as Dr. David Milligan mentioned, we're here really not only helping to solve the immediate traditional or solid organ transplant issues, we want to be of help for all the visionaries and frontier researchers to enable your off-the-shelf 3D printed organs, xenon transplant organs. So instead of building a CGMP facility next to each transplant centers, now let Extherma remove the time of the equation, so you get the best organs to the patient in need on time. Yeah. So we here truly think we have the technology breakthrough building the platform to enable all regenerative medicines. From the cell gene therapy field, actually, we're already in revenue now, we have a product launched and a series of product coming out. Also, we can help on the live vaccines and the engineered natural organs. And last but not least, high throughput drug discovery. And I heard many talks about bringing this new personalized tumors on chip. And this is indeed the part we can be of help and truly solve those time issues, solve the mutation issues. So now the central lab don't need to have 24/7 staff members, and you can preserve the live tumors so you can test the drug on top of it and save patients' life. Yeah, here I want to talk a little bit on the cell gene therapy, and this newly emerged industry actually is very fast approaching to the clinical outcome. So you see autologous cell therapy since 2017, and we see it truly can cure the cancers. But you see biopreservation is indeed not a small piece, actually being involved in multiple steps along the way, 3 to 5 times. And when we move from the autologous to allogeneic, that means we need to build a massive master cell banks, work cell banks, 10 to the 9th, it's a really large productions we need to carry through. So that requires much higher performance of the cryopreservation. And then when we talk about localized injections, so especially talking about the knee injections, the brain injections, or local tumor injections, then toxic components, which is the cryoprotectant, actually could be problematic. But unfortunately, you see, since the 1950s, the DMSO being first discovered to be used for red blood cell preservation, still being used today. And because it's not very effective, we actually need to take aborted animal and the human serum. So you can see the ineffectiveness, and we all notice that for sensitive type of cells, 15% of recoveries, but over the time, you may not end up with any cells left. But the more alarming things on the toxic side, many of the transplant surgeons you know, like the DMSO actually cause a lot of clinical side effects, because vomiting, diarrhea is very common, sometimes can be very lethal. But if you look at molecular level, it's more alarming. Like this Nature paper talk about even just with 0.1% of DMSO in the continuous cell culture can lead to 50% proteomic mutation, 70% transcriptome mutations, 13% epigenetic mutations. So really need to be reconsidered, is this good enough for maintaining your cell therapy's potency? And also, you can see the cause of chromosome damage as well. But on the other side, for process engineer, I know you all know this DMSO is such a painful thing to deal with because it costs a lot of leachable extractables, require you to use a 10 times more expensive bag to carry it. So, and involved with animal serum, just another variation. So we never can have the same cow have the same serum, like make sure they eat the same grass and produce the same thing. So, but unfortunately, FDA, since they wanted to ban DMSO for since 1965, we still not been successful because alternative is just not being effective. But we believe XT Thrive could change this. So this is the first product that we brought into the horizon, actually truly to help scale up production's manufacturing need in cell gene therapy field. It's completely DMSO-free and chemically defined and protein-free, and really giving us the chance to remove the toxic component out of the equation. So we also have a very strong safety data, non-toxic in vivo, and we show indeed that this serves as excipient, remove out of the blood and the system extremely fast and complete. And the non-genome toxic, and giving you not causing micronuclear issues and not causing like all the profound genotoxicity you see from the DMSO. Also, this is really scale-up production friendly and giving us the extension of a pre-freeze incubation time. So now you can have a larger scale GMP product. Also, you extend the post-thaw incubation time, so you don't need to worry about the patient side management and going to infuse into the blood and giving you extended period of time, and no wash necessary. So this is critically important for you to have off-the-shelf, ready-to-use, non-manipulated cell products, and giving us a very excellent liquid handling for large-scale productions and a long shelf life. We have some stability data. So now last, I will just want to take you through some how important for localized injections. Here, we gave like the rats 50 microliters in the pain point of the palm. So just 50 microliter equals to about 12 milliliter for the adult man. And you can see after 30 minutes of post-treatment, compared to the DMSO-based solution, XT Thrive shows no changes compared to the DMSO control. Even with such a small injection, DMSO group, the rat has like a really swollen entire hind limb and extremely painful, does not allow touch, has a fast respiratory reactions. So you really have to rethink, can I really introduce DMSO into my patients for localized injection? So because the time is up, like I will stop here, but I have a lot of data, and I also brought some of our product brochures. If you're interested to know more, like we actually currently have some early access program and be featured on many of those repository articles, and we're looking forward to be off to help solve your manufacturing issues and biopreservation issues. So your success is truly our values here, and we're looking forward to hear more. So here, we're working very hard to bring you more time so you can do your best. Thank you.